Okay, let's talk about this epigenetic process that I've alluded to so much. And here's some objectives. We're going to need to talk about DNA very briefly, scripting and transcription. Define it. This is the reason I didn't start on this section, is because this is not exciting stuff. But we are going to talk about how epigenetics works to change the way DNA does scripting. In essence, why we don't grow ears out of our eyes. That's what I'm going to be talking to you about. So, quick group discussion. Maybe grab a buddy, and this is the age-old question. Ask them, all right. And the reason we're going to talk about both is these two guys, Mendel and Darwin. So Mendel was a monk who worked on, he really created the theory for genetic expression, right? And so a gene is just a set of codons. It's basically a code. It means something in the DNA script, right? So we have the nucleus of the cell. Within the nucleus, there's these chromosomes. And then within the chromosomes, when we unpack it, we have DNA. And a certain codon of the DNA is the gene, right? So he came up with this notion that there was genes that promote heredity, and this is the nature side of things. And then Darwin came along shortly after Mendel and posited that there were evolutionary processes that occur and that influence the way that heredity works. And so we're going to look at where both these theories are now, kind of the state of the art. And there's things that are right or wrong about some of their writing, but we're going to look at where it's brought us, these theories. The first thing is genetics. So we're going to think about the cell as being the basic unit of life, and these are the things that cells can do. And in humans, there's about 50 to 70 trillion cells. And this nucleus contains two chromatin DNA and at least one nucleolus, which manufactures and holds the RNA, which is responsible for taking the information that's in the DNA and producing proteins out of it. So how did we find out about this DNA stuff? Well, it actually comes kind of out of my field, which is radiography and X-ray stuff. You had these two scientists who, prior to 1952, prior to their work, people thought that RNA was the code of life. But Hershey and Chase did an experiment in which they proved that it was actually DNA that was responsible for passing on heredity. And this is their experiment. It's really weird. They took different types of viruses, and they fed them some radioactive stuff. And they either gave them radioactive sugar, which would bind to one part of like the RNA, or they gave them a radioactive, I think it was sulfur, which would bind to the DNA. And then they let them infect a cell, and they blended those cells up, and they figured out what part was radioactive, right? And what they found was that it was actually the DNA that had been communicated, not the RNA. So I want to point that out, that sometimes when we talk about radiation, we think about it just being damaging stuff, or stuff that causes problems with reproduction. Radiation has been here since the beginning of life on this planet, and it's a large part of how we know that DNA is the codon of life. And I think thinking about radiation and the effects of radiation helps us, it gives us an idea of how the fulcrum gets pushed the other direction. If radiation's always been here, but radiation always also hurts babies, then there has to be processes in place that help repair that damage, right? Because interestingly enough, it was also through X-ray diffraction that we found the DNA molecule. So Watson and Crick were using X-ray crystallography along with Maurice Wilkins. And I want to point out that Rosalind Franklin, it's a man's world that we live in, she's the one who actually took the picture, right? She didn't get the credit because she died of cancer. And the Nobel Prize is only given to people who are alive. But in essence, I think she found DNA and the double helix model, because this is the picture that they used to posit the double helix model, and she took it. But they more or less stole the Nobel Prize from her. It wasn't awarded to her. I'll climb down off my soapbox now. So, genes, though, yeah, bless you. chromosomes, when the nucleus divides, the chromatin contracts into rod-shaped bodies. So when we look at the X chromosome or the Y chromosome, what we're looking at is all of the DNA has just been spooled up. It's like a fishing line that got reeled in, and what we're seeing is now the reel of all that fishing line. And we'll call those chromosomes. But gene is just 3 codons of DNA that equals 1 gene. So this is a gene. That's what we're mapping with the Human Genome Project. The environment, let's define that too, because what we've talked about is we're talking about environmental epigenetics. There's an environment in which this is occurring. And these are factors that include stress, nutrition, exposure to toxins, and type of social interactions. And I've said that for the human experiment, these social interactions are particularly important because we have this attachment that we form, and that's a big part of how babies survive. So let's think about the DNA being a script, right? And I'm a big fan of literature. That's what I have my degree in, my master's degree. And so I like the play Hamlet. And I've got, the classic soliloquy from Hamlet, to be or not to be, here on the left side of the page, right? But there's a lot of different ways that this could be read, right? So we have Kenneth Branagh doing to be or not to be, looking at himself in the mirror. This is a really bad Swedish production of Hamlet where he's looking under the stairs for some reason while he's saying it. And you'll notice down here, I don't know if you can see on your PowerPoint, but we have 2 robots and a man who are making fun of him while he does it. This is from Mystery Science Theater 3000. I'm a big fan of that show. And then finally we have Rosencrantz and Guildenstern are dead, right? Are there any theater people who understand anything that I just said? Basically, we have a traditional production of Hamlet, right, which has the actor saying what Shakespeare wrote, right? We have a slightly skewed version of Hamlet where we have Hamlet saying what Shakespeare wrote, but a robot and a man making fun of him while he's saying it, right? And then down here we have 2 people just making fun of things, right? Each one of these processes was built off of this soliloquy, though. So the DNA is capable of being expressed in different ways. Let's talk about how that happens. To do that, we're going to need to think a little bit about what DNA is. So I've said it has a language in it, or it has a code, it has a script. Sometimes we talk about DNA scripting. And it's made up of 2 sugar phosphate chains and then nitrogenous organic bases. And the way that I have my students remember it is with this mnemonic at G and C. It just helps them remember what binds with what. So the adenine binds with the thymine, and the guanine binds with the cytosine. And if you just notice, if they have a Y in them, they're a pyrimidine, right? Nerdy stuff that is helpful if you're taking a class in that. And I want to differentiate it with RNA. So it's deoxyribonucleic acid. That means that it looks like a ladder rung, and it's got both sides of the ladder. This only has one side of the ladder, so it doesn't have the deoxyribonucleic acid. So it's double-stranded. It has thymine, where RNA has uracil. And these are long chains of nucleotides. They're very, very long. In fact, if I had a single strand of your DNA and I said, run in that direction, you'd probably get almost to downtown, right, before we would exhaust all the information that's in that carries information. The RNA has a lot of different purposes, but the purpose we're going to talk about today largely has to do with protein production. So this gives it the script, and the RNA acts out what's in the script. It builds the protein. So it builds the receptor in our case. It builds the glucocorticoid receptor. So thinking about DNA as a script is helpful because if we have the script, if we know what the script is, we can make clones, right? So this is, the first cloned organism was a sheep in Scotland. It was actually named after Dolly Parton. And we have cloned puppies now in Japan, right? You may have seen that in the news. People are cloning their dogs in Japan. And then, of course, we have it in popular culture, like Clone Wars and stuff like that. So epigenetics, though, is not necessarily talking about cloning, right? It's a different way of thinking about it. It's saying, let's alter the script slightly, right? So Epi comes from Greek, which means at or on or upon or over or beside. So think about the EpiPen, right? It acts at or on someone, right? It just means on top of, is maybe the easiest way to think about it. So when there are cases where the genetic code alone isn't enough to describe what's happening, why these people are acting out Shakespeare differently, looking at the script is not enough to tell me why. There's someone who went along and made marks on the script. They wrote on Shakespeare's script and they said, when he says this, do this instead, right? So they altered the script. They took the original script and they wrote over it, right? So now if I take that script and I put it in a Xerox machine, the next time that it's copied, I see those instructions, right? And if I copy the copy, I see those instructions on down the line, right? So those instructions are what I'm after. And the instructions are these modifications to genetic material that change the way either the genes are switched on or switched off. So we looked at earlier glucocorticoid receptor genes. And we said that for a typical nurturing environment, the gene was switched on for protein production of glucocorticoid receptors. So the brain made glucocorticoid receptors. In the presence of stress, there is an epigenetic process that's telling it, don't make those. Change the script. And every time that DNA gets reproduced then, it gives the same script changes, right? So epigenetic modifications are a good thing. They're not a bad thing. They are blueprints over and above the genetic code and on top of it. And they program cells for decades. If we go back to this picture here, this will help us understand why they're good things. So the very first cell that we're going to talk about is the zygote, right? And it is totipotent, which means it could be anything, right? It's almost like sometimes when people talk about God, they talk about being omnipotent, all-powerful. This is the same thing. The zygote is omnipotent. It is all-powerful. It can be all potential cells. It has the eye and the ear and the tongue all in one. But what's going to happen is it's going to roll down this slope, and we call this the epigenetic landscape. As it rolls down the slope, so it might have to go down this little canyon here, and now it can't go and be on this side of the canyon anymore. So it is multipotent. It could still be either of these guys, but it's no longer totipotent. It's not a zygote anymore. As it rolls further down the cell, it becomes unipotent. It only makes B cells. This one only makes macrophages. This one only makes fibroblasts. Okay? So this is precisely what's occurring during human development, right? That as the brain develops, it is going through this exact same process, right? And so what I'm telling you is that this process begins basically shortly after conception. And once we have a brain, so gestational week 4 or gestational week 6, we have the beginnings of epigenetic processes that are going to be communicated on for the rest of that individual's life. And potentially could be heritable. I don't know. That's one thing that researchers are working on right now. So, but the part, I don't want to get lost in the weeds with this. The part that we've looked at is just one thing. I've shown you big picture. I'm not just talking about brains. I'm talking about kidneys. I'm talking about everything. But the one that is most important to us in our understanding of child development is the parts that mediate the HPA axis. Okay, so we've kind of gone full circle now. We're back to this HPA axis of glucocorticoids being secreted from the adrenals and feeding back both into the hippocampus and the hypothalamus as well as into the pituitary and changing the way that these things respond. So ideally, once the stress response has been, the adrenal glands have pumped out their adrenaline, they'll release enough glucocorticoids that those will filter back into the brain and say, okay, we're good, turn it off. Turn it off. But if we have a lack of receptors in the brain, we're not getting the signal to turn it off. So let's go back to environmental epigenetics again. What we're saying is that the environmental events like stress activate intercellular signals that remodel the epigenome of the glucocorticoid receptor. Okay? And these lead to sustained alterations in the structure and function of the genome. I've got a picture of what that looks like, I believe. Oh, I lied.